appreciate the opportunity to introduce our products. Today, I would like to talk about the optical in vivo imaging system. We have 4 products: bioimaging systems, Fluorobox for DNA gel, KB for Western blot imaging, FOBI is for fluorescence in vivo imaging, and Luci is for luminescence in vivo imaging. So, our company's products are imaging devices that use fluorescence and luminescence, and there are in vivo and in vitro products, respectively. Among the 4 products, I will tell you about the optical in vivo imaging system, which can get these image data. These data can also be obtained using devices such as PET or CT. However, it is very difficult to maintain equipment using radiation in a normal laboratory. But, luminescent or fluorescent materials are already widely used in biological lab due to their ease of experiment and safety. They can be easily applied for biological research. In vivo imaging was first developed in hospitals for diagnosing human diseases. Among various imaging technologies, we have specialized in developing optical in vivo imaging systems. Unlike X-ray, PET, or CT instruments, which rely on radiation, optical imaging uses light-based sources, such as fluorescence and bioluminescence, making it much more convenient and accessible for typical biological laboratories. In fact, fluorescence and luminescence experiments have long been common techniques in life science research labs. Optical in vivo imaging can be categorized into luminescence and fluorescence. Both luminescence and fluorescence imaging systems make similar image data. However, because the characteristics of the probes used are different, there is a significant difference between the 2 imaging devices. I would like to explain using the tumorization experiment as an example. There are 2 mice here. One is injected cancer cells containing luciferase for bioluminescence imaging. The other has been injected with green fluorescent protein labeled cell instead of luciferase for fluorescence imaging. To obtain images of cells with luciferase, we inject luciferin into the mouse. Luciferin circulates through the mouse's body and, when it encounters luciferase, emits light through a chemical reaction. This light undergoes various optical phenomena like refraction, reflection, and diffraction. And the light is projected out onto the skin surface. Unlike radiation, light cannot pass through mouse tissue in a straight line, so the light source cannot be seen as itself. Instead, we can capture the light projected onto the skin surface using imaging devices. The light produced by luciferase inside cells is extremely faint, extremely weak. So, that light cannot be seen with the naked eye or even under a microscope. When this weakened light finally exits the mouse tissue, it becomes even dimmer. To capture such faint light, super highly sensitive cameras are required. Therefore, luminescence imaging devices require very high sensitivity camera sensors and cooling to reduce noise. Usually, in order to obtain bioluminescence images, a system that cools to -80 degrees Celsius is required. In contrast, fluorescence produces relatively bright light. Generally, it emits light strong enough to be visible to the naked eye. However, fluorescence imaging involves a slightly different process. To obtain images of GFP fluorescence cells inside the mouse's body, we require an excitation light. Unlike bioluminescence, fluorescent materials cannot generate light on their own, so they need an excitation light. When the excitation light that penetrates the mouse's body excites GFP, GFP absorbs part of the light energy and uses the remaining energy to emit light of a longer wavelength. The emitted light is projected onto the skin surface, and we can get an image of this projected light. By the way, we are interested in the emitted light by GFP, not the excitation light. Therefore, we need optical filters to eliminate the reflected excitation light and allow only the GFP signal light to pass through. In the case of fluorescence, although a considerable amount of light is lost, it initially emits much stronger light compared to bioluminescence. Therefore, even with a low-sensitivity camera, we can get images. So, we need a technology that can filter the excitation light and separates the autofluorescence from signal. In other words, in the case of luminescence, the brightness of the light is weak, but it shows a high signal-to-noise ratio because there is no reflected light or autofluorescence. To capture bioluminescent signals effectively, it requires highly sensitive sensors and cameras with very low temperatures for long exposure times. On the other hand, fluorescence requires a filter optimized for in vivo imaging to remove background. However, if autofluorescence is overcome, the brightness of the light is relatively strong, making imaging possible with low-sensitive camera in a short time exposure. Because of these differences in the characteristics of luminescence and fluorescence, we developed the two devices separately. There are various optical in vivo imaging systems available on the market. Among them, the representative product is called IVIS, which is widely used by many researchers. And many other companies are making products that combine bioluminescence and fluorescence capabilities similar to IVIS. As mentioned earlier, bioluminescence and fluorescence exhibit significant differences in their characteristics. Many difficulties arise when trying to get two types of images from one imaging system. It becomes very complicated, and as a result, it becomes huge, and there is no choice but to focus on luminescence, and the quality of fluorescent images is relatively bad. Because it is complicated, it can be out of order easily and the cost has become higher. So, we make them separately, FOBI and Lucy. If you make them separately like this, the functions of luminescence and fluorescence will be faithful to each other. The device becomes simpler, more compact, easier to use, and high quality for each function. Because the device is simple, maintenance becomes easier, and the price of the device can get lower. The main application of the luminescence in vivo imaging system is tumorization. The primary application of the fluorescence in vivo imaging system is drug tracking. Of course, as long as a luminescent or fluorescent probe can be attached, the system can be applied to a wide range of other research fields as well. First, I will introduce the fluorescence imaging system, FOBI. Unlike other competitors, we used a color camera. Yes, actually, that color cameras generally have lower sensitivity compared to black and white cameras. That means, if the specifications are the same, the sensitivity of black and white camera have more sensitivities than color camera. But there are many different color camera sensors. So, we can choose a color sensor with enough sensitivity for fluorescence in vivo imaging system. Both images represent the same animal. The data of the competitor product on the left does not have color information, so they had to use pseudocolor. Therefore, it is difficult to distinguish between autofluorescence and signal. The image got through our product on the right is the raw data without any processing. You can clearly distinguish between signal and background without any special image processing. In fluorescence imaging, unlike bioluminescence imaging, there's no need long exposure times. Therefore, the imaging speed is fast enough to capture video of the fluorescent signal. These videos are not commonly used in research papers. However, it is of great help to researchers in finding the location of the signal. If the imaging speed is slow, the signal cannot be found quickly. Then they may give up looking for a signal and they will not be able to get any data. However, if the imaging speed is fast, the researchers can easily find the location of the signal by changing the mouse's posture in various ways, so important data is not missed. And the FOBI is incredibly compact. When we first developed, we also made it a little bigger than this. However, most of the test subjects are mice. Moreover, laboratory space is becoming limited these days. There was no reason to make the device large. Although FOBI is small, it can get images of 3 mice at once. Because it focuses only on fluorescence, its structure is simple. The simple structure not only makes it easy to use, but also makes maintenance convenient. FOBI offers 4 selectable channels: blue, green, red, and NIR. You can configure the system by choosing only the channels you need. When selecting fluorescent dyes, it is generally recommended to choose longer wavelengths. Fluorescence with shorter wavelengths has a shallower penetration depth through biological tissues. In addition, autofluorescence tends to occur more strongly at shorter wavelengths, especially on the skin surface. I think image data is the most important for imaging devices. Let's check its performance through a few samples. First, we checked autofluorescence, which is the most problematic factor in fluorescence imaging. This autofluorescence does not depend on the device. Because it is a phenomenon that appears on the surface of the experimental sample, it is a fundamentally unavoidable interference factor. However, the point is how effectively it is removed using optimized filters for in vivo imaging and software. Picture number 1 is bulb C with white hair. As shown in the picture, very strong autofluorescence appears in the hair. Therefore, when getting images of mice with hair, it is better to remove the hair. Picture number 2 is a nude mouse. Even in cases there is no hair, autofluorescence appears due to urine and feces stains. Figure 3 is the autofluorescence of the red channel. This autofluorescence comes from the stomach and intestines. And it comes from digestive juices. Therefore, this autofluorescence must be taken into consideration when analyzing the fluorescence of the abdomen. Figure 4 shows autofluorescence present in plant leaves. There is very strong red autofluorescence in the chlorophyll. The first application of fluorescence in vivo imaging is tumorization. GFP is transfected to tumor cells to make a stable cell line and then tested on mice. In the past, GFP was widely used, but since GFP with a short wavelength has poor tissue penetration, the use of long-wavelength IRFP has increased recently. The second application is stem cell and immune cell tracking. Transfecting fluorescent genes using lentiviruses can alter the characteristics of stem cells. Therefore, when labeling stem cells or immune cells with fluorescence, chemical dyes are preferred over genes. By labeling the cell surface or cytoplasm with fluorescence, it becomes possible to track their movement within the mouse. Therefore, it is used in cell therapy research. The third application is the drug delivery system. Particularly, you can label and track peptide or antibody drugs targeting cancer cells with fluorescence. Fluorescent substances like ICG or Cy7, which offer excellent tissue penetration, are used in such cases. In vivo imaging may be difficult some cases. If the signal is weak due to optical limitations, it may be can't to get images with the live mouse. This issue is more related to the limitations of optical imaging rather than the imaging device. In such cases, it's possible to get images ex vivo by extracting tissues from the mice after the experiment is completed. Furthermore, fluorescence imaging can be applied to various applications on the same animal. In the image, green represents cancer cells, while red represents stem cells. The stem cells have been modified to have the function of inhibiting cancer cells. Tracking was performed in vivo as shown on the left, and then reconfirmed ex vivo as shown in the upper right image. And we can check it again by slicing it as shown below on the right. In these images, it is evident that stem cells have inhibited the growth of cancer cells. Sometimes, we can obtain data from microtubes or well plates as shown in the image. These in vitro data are used in papers by images and quantitative values in tables. Sometimes, although it may not be used in papers, but it can be used to verify whether cells or drugs have been effectively labeled. Another application is the expression of fluorescent genes in plants. This image shows a specific gene inserted into rice. When inserting a specific gene, GFP is also included to assess the level of expression through fluorescence imaging. Then, only the rice containing the gene can be collected and tested. Without such imaging equipment, experiments can become complex and time-consuming. To confirm inserted gene, we need to plant these rice, germinate them, collect the leaves, and check through PCR. After confirmed, it is harvested, and then experiment materials are prepared. But that would take one generation of plants just to prepare the experiment. Next is fluorescence imaging from plant leaves. You can get images of the expression of specific genes in plant leaves. The paper represents the performance of the device. We have been used in over 400 papers. Using FOBI makes in vivo imaging easier. Next is the bioluminescence imaging system, LUCI. Like fluorescence, there are also many competing products for the luminescence imaging on the market. However, IVIS occupies most of the market. What are the most important specifications, and why does IVIS have a high market share? Most important thing for luminescence imaging is sensitivity. The light of fireflies appears very bright, but if the gene is transfected into cells for expression, it is too weak to be seen with the naked eye. As mentioned earlier, as this faint light passes through animal tissues, various optical phenomena occur, resulting in the projection of very weak light onto the skin surface. So, bioluminescence imaging requires an ultra-super-sensitive optical sensor. We used a back-illuminated EMCCD sensor. This sensor can detect as few as 8 photons and make clear image data. LUCI incorporates such a sensor. We excluded parts that were unrelated to the performance, the sensitivity, and focused only on high-sensitivity images. This approach allowed us to develop the product that is highly sensitive and fast, while also being simple, compact, and user-friendly. Furthermore, we were able to maintain competitive pricing. The data presented here is a comparison with the IVIS using the same sample. As you can see in the images in table, there is only a 5% difference. LUCI not only has good sensitivity, but also provides excellent imaging data like IVIS. So, as shown in the picture on the left, we were able to get images of the cancer planted on the mouse's leg and the lung. In case of the lungs, there are many obstacles for light to pass through. The chest muscles, breastbone, and numerous membranes block the transmission of light. As you know, when light passes from one material to another, a great deal of light is lost. So, it is difficult to get lung images. As you can see in the picture, a clear image of the cancer that has spread to the lungs was obtained using LUCI. Bioluminescence can be applied not only in vivo, but also in vitro, such as plate as shown in the picture on the upper right. It is also useful for checking gene expression in plants, such as Arabidopsis in the picture below right. LUCI is designed to get images quickly. When the exposure time is set to 1 second, our competitors often require tens of seconds to over a minute to capture the images. However, with the LUCI, you can capture both of normal and signal images and merged images in just 5 seconds. Using LUCI, bioluminescence imaging can be easier. Today, I showed you 2 in vivo imaging systems that can get fluorescence and luminescence images. Other competitors get these 2 images with one device. So, they are more complex, difficult, larger, and more expensive. However, by making these 2 products separately, we were able to get imaging systems that have excellent performance, are small, convenient, and are price competitive. I hope that you would generate high-quality in vivo imaging data and research papers with FOBI and LUCI. Thank you for your attention. We look forward to continuing to contribute to scientific discovery together. Thank you.